portion of the recording of the webinar with Dr. Awad was cut off just the first couple of minutes. So I'm taking this opportunity to provide an introduction and summarize the very early part of what he spoke about during this webinar. The topic of the webinar is big picture updates on guidelines, trials, and biomarkers. And we have with us neurosurgeon Dr. Issam Awad. Dr. Awad is the Chair Emeritus of the Alliance to Cure Scientific Advisory Board. He's the Medical Director of the CCM Center of Excellence at the University of Chicago. And also at the University of Chicago, he's the John Harper Seeley Professor of Neurological Surgery. He's a professor of neurology, and he's a professor in their Neuroscience Institute. The webinar began with news about an update to the Alliance to Cure CCM Clinical Care Consensus Guidelines. Now, the Clinical Care Consensus Guidelines offer recommendations for the standard of care for CCM patients. Developing the guidelines is a project of the Alliance to Cure's Medical Advisory Board, along with invited experts. The original guidelines were published in 2017 and are currently undergoing an update. Clinical care consensus guidelines are developed using a very strict protocol, which starts with an extensive review of the medical literature. Recommendations in the guidelines are based on peer-reviewed evidence, not on personal opinions or experiences or case studies. And the evidence found in academic papers is evaluated for quality. Not every journal article is weighed the same way. We join Dr. Awad in the webinar as he explains classes of evidence and how our experts use these to identify papers with the strongest level of evidence for recommendations.
Everything goes on PubMed, and then you don't know if the two papers are, as believable or whatever. So the classes of evidence has to do with how the study was done and whether it was controlled, and the bias control and the duplicative evidence and so forth is important. And then there is the class of evidence, which also has to do with whether this weighs in favor of doing something or against doing something. And between the level and the class, we can weigh the different papers. And the other important thing about the process that has emerged in recent years, which we are committed to follow, is to not to pick the papers we like and ignore the papers we don't like, because there's always this issue, when you got 2-3 people there, they remember a paper. It's not that they want to exclude others, but they remember something and that's what they quote, and then they may ignore something that's actually transformative in the field or very important, like a paper from the epilepsy literature that is very relevant, that is a clinical trial. And if they don't mention that, then you miss that. So because of that, we go through a mining of the literature and get all the papers that have been published during a given period. In this case, it's kind of easy because it's not from forever till now. It's from the last time that we did the guidelines till now, which is about 7 or 8 years worth of literature. And there have been a lot of papers published. CCM has flourished. And then we divide them by topics, and then we have criteria for which papers must be cited. So the papers that have to do with clinical trials, with controlled studies, or with systematic reviews of the literature must be considered, and the others are provided to the writers. And then we divide the group into writing groups that has the leading experts in surgery, radiation, genetics, imaging, etc. And they go at it, and they take the evidence that was written in the last guidelines, and then the goal is to update it. It's not to rehash the same stuff. In fact, they hate it when Connie and I go back to them and tell them, no, you can't cite that paper from 1958. You know, that's already been cited, and it's great, and it's an important paper, but that's not what the guidelines are about. The guidelines is about new stuff since the Akers et al. Paper that was published in 2017. So the idea now is to, what do we do with this evidence? So we have gone through it. The writing groups have finished putting together their material, and based on their material, they either revised the prior recommendations or they added the recommendations. And there are a lot of new areas you didn't see before, like spinal AVM, like spinal CCM, and like criteria for diagnosis of CCM and so forth that weren't part of the prior guidelines. There is a lot on laser interstitial therapy, which wasn't available before, and various other innovations. And we are now in the process of circulating it through what we call a Delphi consensus process. Delphi goes back to the famous temple in Greece where wisdom was generated, and the idea here is that the opinions by the writers get circulated to all the other writing group, and anybody who doesn't like something about the recommendations or think they're an overkill or an understatement, they have anonymously the ability to tell their concern without knowing who is the one objective. So we are not intimidated by Connie, you know, and others. So the idea here is to be able to say your opinion, and then it goes back till everybody agrees that the evidence is what we are writing. So this isn't about how you feel, it's about what the evidence in the literature says. And it's going to have a lot of stuff on vitamin D that we didn't have before, on aspirin that we didn't have before, and so forth. So it's really good stuff. And the usefulness is the last thing I want to say. The usefulness, I cannot understate the difference this paper has made in the whole body of literature, because when there is a doctor you know, that sees a patient, they haven't seen a cavernoma in 3 or 4 years, they start looking it up on Wikipedia and thinking what they're going to do. Yeah, I tell them to cut through the chase and just go directly to the guidelines. And often my clinical note, when I see a patient and I want to send a note to the doctor, I usually give them the link, the internet link to that, so that if they really don't know where I came up with the idea that you should take vitamin D or not, or whatever, they can go back and check the guidelines, or that it's safe to be on aspirin or whatever. So we're looking forward to having these guidelines updated. And I want to just open it now for any questions just specifically about the guidelines, Connie. Is this kind of the overview you wanted?
Yes, it is. And I know we have 2 questions. Are the guidelines global or US only? And the reviewers who look at it, are they global?
Yeah, so I think we've occasionally had some individuals from outside. I don't know if Rustam is involved this time or not. It's mostly US based in terms of the practice opinions. And there is a very simple reason for this. It's because a lot of the practice guidelines, say in the United Kingdom, are weighed by the National Health Service and what is allowed there and what is not, and so forth. So we wanted it to be useful, and we follow things like the American Heart Association and the American Academy of Neurology style of writing, which are all American focused, if you would, American and Canadian.
Okay. And then my personal question is, could you just mention the new section that we have in this that has been added about diagnosis?
Yeah, I don't want to, tweet the horn too much because it's a short section, but basically there is an area on the imaging which was mostly when should imaging be done, and what kind of imaging, and what's the best MRI for CCM, and when should you do a CAT scan or MRI. And those are still there, and we updated them with new things like QSM and functional imaging and all of that. However, it never addressed the very simple question that, you know, what is it on MRI that makes you call it a cavernous malformation? And what, you know, is there a feature that is diagnostic? How do we establish a diagnosis? And of course, the genetics is another aspect. Sometimes the disease is diagnosed by genetic testing before an MRI is done, and therefore we combine the areas between the two and have a very short, but I suspect a useful section on what are the diagnostic criteria for CCM.
Yeah. And that's something that we're frequently asked. Is there a place where folks can find the diagnostic, where doctors can find the diagnostic criteria? Now, there will be such a place. Someone has asked, and again, let me back up one second and say, please be sure to use the Q&A for your questions for those who entered late and didn't hear me say that the first time. And it looks like everybody's doing that so far. Someone asked, and she's sorry if she missed this, but are we able to access the guidelines? And I can answer that and say that we have the current guidelines, the ones published in 2017, available on our website. If you look in the section, for sure they're in the section that's labeled for professionals. There's a direct link to them.
Or you can go to alliancetocure.org/CCM guidelines.
That too.
Because I use that
You do that all the time. And the new ones, though, the question is, when will we see these published and where do we look?
Yeah. So there are two formats of these. One of them is the complete guidelines, and one is the synopsis guidelines. The complete guidelines cannot really be published because in a journal, because the peer-reviewed journals have limitations on 6,000 words for the article and the number of papers you can quote, which we may exceed. So we will have the full version. It's not unfiltered. It is the same standards of rigor, and that will be posted. It's our material. The Alliance to Cure has the copyright, and they will go ahead and post that on the Alliance website. That's where you get it. That's where you go to get the guidelines. When the updated ones are there, that's when they will be there the minute they're published. Now, the publication format is going to be called synopsis guidelines, and made enough to fit in a journal article. The nice thing about the synopsis is that it will be peer-reviewed, so it's not just the folks who wrote it, but it will go and get critiqued and reviewed by reviewers like we do in peer-reviewed literature journals. And they will look and see if we were biased, because, you know, we are the CCM community, and they will point some things. I think we're pretty confident that we're following the most rigorous standards there, but you know, peer review will slow it down a little bit, and then that version will be published in a journal. And we haven't gotten this far, but normally we will find a way to purchase the open access or to negotiate the open access to that, so you don't have to pay to get that article. You'll be able to get it, even the synopsis, which many doctors like to read the synopsis. So you'll have two versions, one online and one that is the synopsis that is peer-reviewed. Okay.
And then we have a couple questions about what will be covered. One is, will it cover the use of antithrombotics? And someone else said, will it include the type of MRI machine? Can you?
Yeah, I mean, there hasn't been a lot of change in the latter, in the issue of the MRI machine. We, in order to get a comparable count, you need to have the same type of machine. We normally recommend the 3 Tesla if you're counting lesions. If you're going to look at the size of the lesion and whether it bled, a 1.5 Tesla is fine, but 3 Tesla is recommended if you want to count lesions accurately and compare counts. So you don't say, oh, I had 20 and they told me I had 18 and whatever. So it has to be done the same way, and we do mention those things. There are also certain machines that are used for things like functional MRI and so forth. So we will mention all of that. So yes. And what was the second half? Sorry, this is the machine. The other one?
Antithrombotics.
Yeah, the antithrombotics takes a big portion of some of the changes. I think we have more evidence of safety of antithrombotics than the first time that we wrote about it. There is not going to be evidence of benefit of antithrombotic because that evidence is just not there. And when you go, you hear a lot of people with opinions, but when you really get to the science of it, the evidence that you get a benefit from antithrombotic isn't there, or even the evidence that they are safe after you bleed is not there. So nobody's telling you to go take aspirin after a bleed so you don't bleed again. It is the issue of if you have to take aspirin for your heart and your CCM has not bled recently as a symptomatic hemorrhage, then we have a lot of evidence that you should do that to protect your heart and do it. And there may be some hint of a benefit for CCM to be tested in the future.
And the last question on this, how do we get our PCPs to read the guidelines?
Well, that's going to have to be, it's a PR challenge. And, there may be ways of sending mass emails. We have to buy the email lists of some people and send it to them. I think it's going to be good to have that. And as doctors who take care of CCMs at the CCM centers, in fact, we accredit centers based on the guidelines. So basically, when we screen a center, at least that's how we used to do it when I was doing it, is that basically if they know about the guidelines and are following them, then they become a CCM center. If they don't, we're not going to be excited about getting them in there. So they have then a role to tell their referring doctors. So you go see center A or B in your community, and that's an accredited CCM center. Hopefully we can tell all our CCM center consultants, and that's something Connie can do, please, in your consult notes, in your whatever, just refer to the guidelines so the doctors don't think you're shooting from the hip.
And then those notes go back to the PCPs as well.
Yeah.
So they can see there that that's what they should be looking at.
Right.
It often is better if it comes from a peer than from a patient.
Exactly.
All right.
Also, don't hesitate. Let me just say one last thing. Don't hesitate to be an activist. If you've got a doctor that doesn't seem to have a clue, you give them a copy of the guidelines.
Yep.
Or a link. All right. Very good.
Right. So now you were planning to share with us information about trial readiness and about the clinical trials that are underway and planned.
Yeah. So a lot of exciting things the last year or so. So as you probably already know, the Atorvastatin trial, which was aimed to enroll patients who had bled within a year as a symptomatic hemorrhage. And the goal is to see whether using Atorvastatin at 80 milligrams a day, which is the high dose for stroke prevention, is able to prevent bleeding in the cavernoma when used over a two-year period, counting the bleeds during a 2-year period. And half the patients took placebo, half the patients took Atorvastatin. The safety committee did not stop the study. So the reports that they shared with us was that the complications and symptoms were about the same in the placebo group as in the statin, including stuff you might think only happens in statin, like, I feel foggy or my leg is hurting or whatever. There were enough people in the sugar pill group that had those same symptoms. symptoms. So anyway, we will count all of that once we break the blind. Right now, the only people who looked at breaking the blind, meaning who is on drug, who is on placebo, were the safety folks, and the final patient will complete their 2-year visit in early July, I believe, late June or early July. We are preparing the data sets so that we can very, very quickly analyze the primary and secondary outcomes. So let me tell you what the primary and secondary outcomes are. So this is the first trial that is powered, meaning it has enough patients that we can count the amount of bleeding in the lesions as measured on QSM. And I'll talk a little bit later about how this has evolved. But QSM is a way of doing an MRI to measure the amount of bleeding in the cavernoma, not just to say, is it bigger or did it bleed recently, but to actually look at the amount that it bled. And we measured the QSM 3 times in the patients. We did it at the time they enrolled, they started to take the drug, and at 1 year and at 2 years. And then we're looking at changes in QSM, increase or decrease at these 2-year intervals. So we call them a 1-year epoch. So each patient had 2 of those epochs to contribute to the study. And the changes in QSM is as simple as if it got greater, that means you're bleeding more in the lesion. If it got smaller, that means the blood is going away in the lesion and you're not bleeding. So the idea here is we have enough patients, with 80 patients in the trial, to be able to tell you whether that is beneficial, harmful, because we did it as a two-tailed study, so we can tell if statin is making you bleed more, or if, despite this very sensitive biomarker, we can't tell a difference between the groups. So the answer will be one of those 3. There is no other surprises, and that's the primary outcome. The secondary outcome is the actual bleeds, meaning the number of bleeds that the patient had, and we count all of those. And people who bled and went to surgery, we count that. So it's a very rigorous way of doing it. There are more bleeds in this trial than in the Italian study and in the recursion trial, because we enrolled specifically people who bled in the prior year. So, and we had about half of the patients brainstem lesions. So we have a lot of high-risk lesions that were enrolled, and therefore we'll have enough bleeds to be able to compare. And that's the secondary outcome. There is another secondary outcome, which is the leakage in the cavernoma as measured with the gadolinium permeability. So the idea here is the lesion becoming less leaky, and we will be able to understand that a little bit. That's going to be important for other drugs, the lessons learned there. So a great study, and I think it'll be exciting whatever the results are. And the one important thing for the community to know is that we cannot change the label of Atorvastatin based on the results of the study, no matter how strong they are. So if it shows more bleeding, we cannot put a label on Atorvastatin causes more bleeding, or it causes less bleeding, therefore take it. The reason for that is because the trial was done very specifically under what they call an investigational drug exemption, which one of the criteria they give you to allow you to use that drug is that it's a previously approved drug for another indication, and you're probing the likelihood. So that's why it's called AdCash epoch. So that stands for Atorvastatin treatment for cavernous angioma with symptomatic hemorrhage, exploratory proof of concept. That's what epoch stands for. So if we have a positive concept, then we can go to the FDA and propose another study, or see what other evidence they will need to change the label. But that won't stop us from telling you the results. And I suspect if it's a strong benefit, since this is an approved drug, many people will use it. So we'll see. We'll see where this goes. So that's Atorvastatin. So the other project that actually got completed, so Atorva will not be finished and reported till this coming year, 2024. But what we completed in 2023 was the Trial Readiness Study, which is another large study, $5 million funded by the National Institutes of Health, which was designed to include centers, different places in the country. So the Atorvastatin trial was only done in Chicago, but this included sites throughout the country. They were the early centers of excellence at cavernous angioma centers of excellence. And basically the idea there is to inform the community and the pharma and everybody who's planning trials in the future, what are the event rates? So if you see 200 patients with cavernoma at the Barrow Institute or at Stanford or in Chicago, how many are eligible for a trial? And if you follow them, how many will have events and at what rates and so forth? You'd think that that's something easy to get, but that's very presumptuous because there is a lot of evidence out there that is not collected this way, and a lot of mistakes can happen. So in fact, some of the big volume centers contribute very little patients to medicine trials because they take all the patients to surgery, or the patients are referred to them to surgery. While other centers like Mayo Clinic and University of Chicago, New Mexico, the patients go there for various reasons, medicine and surgery, and therefore you have a different rate. It turns out that about 1 out of 6 patients with cavernous malformation presenting to a center are potentially eligible for a trial. The others either hadn't bled recently or didn't or are going to surgery and therefore will not, or had some other exclusion criteria like they can't have MRIs or so forth. There is another interesting thing about this. What patients are followed and how that may change a little bit, of course, if you end up having a drug that you're testing. And also it will change a little bit if the inclusion-exclusion criteria were different. So for example, the Recursion trial that finished enrolling patients this past year, enrolled 60 patients. It was symptomatic cavernous malformation. It didn't say you had to have bled from a cavernoma in the prior year. So, the reason why one would do this trial or that trial, the inclusion-exclusion criteria, depends on what type of population you think your drug is most likely to have an effect in. And if you overshoot that and you are over-ambitious, your trial is going to fail because you won't have enough events to show a difference. So if you include everybody with CCM and the majority don't have any events, then how are you going to tell that the drug is better if without the drug you don't have many events? So you want to bring in the people that are likely to have problems, because those are the ones where you can count events, and also are the ones that will benefit from an experimental drug. So that's the kind of balance between these. And I think it's going to settle somewhere between symptomatic, attributable to the lesion within 1 or 2 or 3 years and whatever. And that's how the different drug companies are going about it. They also don't want to be too focused because, they don't want to have to do 2 different trials for their drug, one for inclusion within a year and one within 2 years. So it's a balance between being able to show benefit and being able to also benefit as many people as possible. So that CASH trial readiness, we completed the follow-up, and we found out that about 11 to 12% of the patients will have a symptomatic rebleed. These are people who bled in the prior year, did not have surgery. So about 11 to 12% of them will have a bleed in the first year, and another 11 to 12% will have in the second year. So it didn't look like the rebleed rate went down in the second year. It stayed about the same. It gave us an estimate of about more than 20% of the patients will bleed within 2 years. So that is a known thing based on which we can build clinical trial models rather than going in the dark. You know, the people who started the trials, like the Italian group, did it in the dark because they didn't have this data, so they couldn't power the trial to detect a difference. The other thing we showed is that QSM, which is the measurement of iron in the lesions, increased more frequently than clinical bleeds, which means that it gives us an opportunity to look at bleeding in the lesions, even in patients who didn't have symptoms. So you can do the trial with fewer patients like that. If you have to count bleeds, you have to have a lot larger number of patients in the trial. And the differences with QSM over 2 years were such that the trial modeling is very, very favorable, and the FDA has accepted this method as a potential method to even accredit a drug. So it's a very big hump that we got over because the data was very good, and the data also showed that every single person who bled had an increase in QSM. So QSM was not a false negative. So it was always there when you bled, but also was there in more patients, therefore you could count it. So I think drug companies now are going to look at their strategy and say, does it make sense for us to have our first proof of benefit with using QSM, with incorporating QSM, or without it? And drug companies will reach different conclusions on this regard. But I think the work funded by the NIH between the two trials is about $10 million, is like a gift to the CCM community, because now there is a huge amount of data and new tools to be used. We measured permeability, and permeability change also increased when the lesion bled. But permeability is not going to be used in trials because the variation between patients is so big that you actually, although the one that bleeds will have an increase in permeability, to have an effect of a drug on that change, there is enough variability between lesions that the number of lesions needed will be not very favorable. So QSM was tight. The permeability was more variable, and we didn't know that before. Also, you'll be relieved because QSM doesn't need gadolinium. So that was good. The other one, had gadolinium dye. Now we continue to use it for clinical scans, but for trial imaging, you're not going to need gadolinium. So that's all important data that we have. And in terms of the big trials, the Italian group published the propranolol results, which you all know that basically there was no significant benefit. There was maybe a hint of benefit, but not enough to be able to define a dose to test in a phase 3 trial. So a little bit more work is going to need to be done. And I think they're working with the European Commission to see if a larger propranolol trial will be done. They also did only familial cases, and there may have been a problem with that. They didn't have recent bleeds, therefore they had only 2 bleeds in the treated group and 2 bleeds in the untreated group. So you can flip your coin and you'll get that. It's a very, very low event rates. But we learned from that, and the next trials will be different. Recursion had a different trial where their goal was mostly focused on safety and tolerability of their drug, and they're looking for some signal effects on benefit. But the trial, admittedly, upfront, out of the gate, was not powered to look at effectiveness. And we'll see what comes out of that, and it's all done in steps. So obviously, a safe drug is valued at much more and will be much more welcome in the community to be tested later. The follow-up to the statin story is going to be the ROCK2 inhibitors, which are molecules that have been studied for more than 7-8 years in CCM in mice. and basically it is a brain targeted ROCK inhibitor, while statin inhibits ROCK throughout the body and has other effects. So these drugs do not lower cholesterol, are not designed for statin type thing. They are designed to alter permeability, improve permeability of endothelium more in the brain than elsewhere. So obviously they're more targeted drugs. Whether that's going to be better or not, we'll await trials, but all of us are very excited about it. And we're also very excited because the pharma that is powered by venture capital is investing huge amount of monies in that beyond what NIH can do. And there are two of these that will probably come to trials in the next year. So I think this is all exciting times, and let's take some questions based on that. The only thing I'm going to ask you, Connie, is to leave me 10 minutes at the end for the plasma biomarkers.
Absolutely. I have a ton of questions though. So just going back a moment to some basics, you talked a lot about QSM. Can you explain what QSM is?
Yeah. So, you know, where you've seen the MRI that shows the lesion to be dark on what they call susceptibility images, meaning the blood picture. It's like a blotch of ink.
Mm-hmm.
And the problem is that that doesn't tell you how much ink there is in that blood. So QSM is about measuring how dark is that ink. So it changes it from a black picture to a gray scale. And every part of the cavernoma contributes a signal. So it can have a part that has a lot of iron, part less. So we measure the average lesional iron content, and this has been validated to be the true iron content in lesions. When we took lesions from patients' heads, they had exactly the amount of iron that QSM predicted. So it's a very accurate, or the FDA would not have bought it. So this was very accurate, very, very clearly done. And so basically we're measuring how much bleeding the CCM lesion has had. We don't know about whether it measures very old bleeding. We know that lesions that bled more and more have darker, more QSM, but some of the stable lesions, their QSM remains the same or decreases very slightly over time. But every lesion that bleeds will have an increase in QSM.
When will the Atorva trial results be available to the public?
Yeah, so, you know, they're not going to be reported at the CCM meeting in July because it'll be just too early. So we're looking at analyzing them in the summer and whether they get reported at one of the meetings in the fall or at the International Stroke Conference in February of next year.
All right. What do you think? There is a doctor, and I'm not sure I'm allowed to say this out loud. There's a doctor who is trying a combination of atorvastatin and propranolol for a few patients, and the results seem promising. What do you have an opinion on this? I think you're aware of this.
Yeah, I mean, the idea of synergy is not wrong. You know, there is a lot of, you know, in chemotherapies, we give two different combined chemotherapies and they do better than each. Whether two drugs are doing the effect of one or are adding, and I'm not sure a handful of cases can prove anything. I think it's good that they're not causing harm. That's about the only thing you can tell. But that doesn't necessarily mean that we would endorse doing this approach because we still don't know how safe statin is. And you may be thinking you're helping, but you may in fact be harming patients or misleading them by thinking they're doing something good when you're not, you know.
Okay. Will participants in the atorvastatin trial, or when will they learn if they were on placebo or drug?
Yeah, so after the results are published, we will work with our IRB to be able to let everybody know what they were on. the implications of that on whether they should go back on the drug or not go back on the drug will probably not be evident, because even if you had a benefit during 2 years by statin or harm by statin, that doesn't mean that doing it 2 years later is going to be effective, you know? So I think the only thing we would have shown is that a lesion that bled in the prior year benefited for up to 2 years.
Okay. All right. Switching to the trial readiness. Regarding the 20% rebleed rate, were there any obvious correlations like the gene involved, the sex, the location of the lesion, vitamin D level, comorbidities, etc.?
Yeah, so yes, but they're the usual suspects that we know about. So no new discoveries there. So brainstem lesions bled a little bit more, but not as much as we had thought. We had thought brainstem lesions are going to be like knock your socks off, bleed more. They didn't actually, and that surprised us. And what that means is that after maybe the first bleed is more likely to be the brainstem, but the brainstem lesion is not necessarily more likely to rebleed. So that was a very important finding. Otherwise, there were really no big differences among the genotypes, familial versus sporadic, or when they presented during the year prior, because, you know, we took people that bled in a whole year before the trial. So some people bled the month before, some people bled 11 months before. That didn't affect. So that basically tells us that the rebleed risk remains about constant for up to 3 years.
All right. Someone's asking about QSM and spinal cavernomas. Can it also predict the bleeding risk or rate of spinal cavernous malformation?
Well, it doesn't do it in the brain, so there's no way it's going to do it in the spine. I never said that QSM predicts bleeding in the brain. So QSM is used to monitor whether a change in QSM is affected by drugs. That's all it's being used to. Any other implied benefit of QSM is not there. That's why I don't tell you, hey, your lesion has 110 or 120. We don't. That's not the application. Now, the spine has another problem with QSM, is the fact that there is bone surrounding the spinal cord. And the lesions that are very close to bone or surrounded by bone, you cannot do accurate QSM, and you cannot even do accurate susceptibility images because the whole spinal cord would look black. So the counts of lesion in the spinal cords are a little bit different than the brain because it's a very narrow, it's as big as my fifth finger and surrounded by bone. So no QSM and no SWI for the spine. So you usually do these T2 or T2 star enhanced images. And the guidelines are going to say that, you know, we don't count lesions in the spine because they don't change what we do. We only go after lesions in the spine if they are causing symptoms.
Okay. Another question about atorvastatin. When you're talking about atorvastatin for CCM, is that the same drug as the atorvastatin that's being used?
Exactly. Lipitor, very cheap.
All right. And then I think we had a pre-question that was, what do we think atorvastatin is doing? Lowering cholesterol and inhibiting ROC? You
yes
answered that
so,
during.
Yeah, so we spent a good part of 10 years clarifying this. And, you know, there is something that was discovered in the heart literature, which they've been using statins far longer than we have in the brain, is that cardiac protection from statin is actually unrelated to the cholesterol. So of course you get a benefit from lowering cholesterol, but that's not the only benefit of statin. So people get on statin to prevent a heart attack even when their cholesterol is low. And then a big study called the Sparkle trial showed that for preventing strokes, statins are beneficial at high dose regardless of your cholesterol level. So you start with low cholesterol, you still get benefit in terms of stroke prevention. So what the hell is statin doing? So what statin is doing is what they call pleiotropic effects, meaning other effects than cholesterol lowering. And one of those effects happens to be Rho kinase inhibition. And that only happens at high dose of statin. So for Atorva, you have to take between 40 and 80, but the robust effect is at 80. Simvastatin, up to the doses approved by the FDA, does not cause enough ROCK inhibition. That's why simvastatin is not being pursued. Now, the stronger statins like rosuvastatin and whatever will be good because they have equal or greater strength than atorvastatin. So in statin, basically what you're doing is trying to inhibit ROCK with a drug that does that and other things. And whether it's doing harm with the other things or everything is helping, we tested it very carefully in mice. And we found a lot of benefit in mice, and that's why we were allowed to do the trial. So it wasn't harming the mice, and it was causing less bleeding in the CCM at the doses we are testing in humans. Of course, not the same dose in the mice, it's the dose per weight of the mouse on surface area. But that's kind of how that works. Now, that's where the advantage of the ROCK2 inhibitors are going to come, right? So the ROCK2 inhibitors are designed to be focused more on the signaling we want to change that is beneficial in CCM with less of the other side effects.
Okay. And then sort of moving into the next implication, can you talk a little bit about any new findings that there might be regarding inflammation and CCM? And also other folks have had questions about anything new with the microbiome research, CCM, and why don't we have more focus on diet than on drugs?
Yeah, let me answer the second one first, because microbiome is becoming extremely popular. You're finding microbiome effect in everything, migraine, Alzheimer's, etc. There are two problems with microbiome research. It's better to explain the effect than it is to change it. And the reason that's the case is because all the studies that detect the microbiome effect, let me take a minute to explain to you how the science is done. We take the stools, you know that, many of you have donated, and then we look at the group that has CCM or doesn't, or the group that bled and the group that doesn't. And in the group of 10, 20, 30 patients each, we compare the ecology of the bacteria, meaning the genes that the bacteria are expressing. So we do the DNA of the feces, and from that DNA, we can extrapolate what bugs are bigger or less or more of them and so forth. That type of science can only be derived in group studies. It cannot be derived in your stool specimen alone. So if I took your stool specimen alone, I cannot tell you if you have a good or bad microbiome. I can't. Even the genes that I find in that, I can't, because it has to be compared in differential gene. So what that means is that we can explain a lot of things and do very elegant mechanistic studies, but it's very hard to change it. Now, people have done fecal transplantation in some diseases to try to affect it. That's going to be very hard to really convince a lot of people to do when we don't know. Now, so we end up going indirect here. We try to say, what are the things that can cause the bad microbiome, or what are the things that can cause the permissive or the bad microbiome versus the protective microbiome. And we have some good ideas on that people can apply in their own lives. You've heard a lot about the avoidance of preservatives in foods and things like that. Treating gut inflammation, if there is gut inflammation. So if a patient has inflammatory bowel disease and CCM, if you treat inflammatory bowel disease, you're going to help the CCM. So And we've shown that in mice. So that type of stuff is an indirect way for us to give you some advice. But the way it's going to happen in the future is going to be that we're going to have some blood tests that reflect the good or bad microbiome. And we're doing a lot of research on that. So if you've got a bad microbiome, there'll be a substance or two in the blood that can be measured in you only. It's like your blood sugar level or your cholesterol level. And we'll be able to tell you the good cholesterol, bad cholesterol, and sugar. When you combine them together, you've got the bad gut for CCM. So that's where that research is going. Once we have that tool, then we can start manipulating. We can say, you know, does vitamin D help you through the stools or another way? Or can I eat more sauerkraut or less sauerkraut, you know, and stuff like that. So that's where we'll go. Until then, we're very cautious in how we apply the microbiome. Now, the idea about inflammation is yes and yes and yes, so that in every study that we have, inflammation drives CCM. And that happens even in the earthworm that doesn't even have lymphocytes or inflammatory cells. They have genes of inflammation, and those genes are whopping when the CCM is active. And in every study we have done, and a lot of our biomarkers in the plasma and so forth are looking at inflammatory markers that are also markers of CCM disease activity. And that is why vitamin D is a good thing, because vitamin D is an anti-inflammatory vitamin. So people who have low vitamin D have a pro-inflammatory states, and that is one of the factors. So yes, inflammation is very important, and it's a big part of what we are studying. Sorry, you're muted.
I know. No, I'm not. You asked me to leave you a few minutes for the biomarker talk, the plasma biomarkers, but can I ask you just really quick the question
Sure.
about what do we think is going on with familial cases who are negative for the CCM 1, 2, or 3?
Yeah, that's a very, very good question, getting a lot of interest. There was a recent paper in Nature from the Harvard group that found what they call a loss of heterozygosity, meaning it's a type of gene disruption that is not detected on regular genetic tests, and it was present in the CCM2 gene. So it's not a new gene, it's not CCM4 or anything like that. It's a way of detecting a variant of CCM2, at least, that is presumed in 2 patients to have occurred because of this loss of heterozygosity. There are a lot of patients now participating in those studies. So loss of heterozygosity, we also, Dr. Marshuk and I had a paper this past year in Nature Communication showing that in all 3 genes, there can be loss of heterozygosity, which means the 2 genes are not equally expressed. That's what loss of homozygosity. So when the 2 genes, 2 copies of the chromosome are singing the same way, that's homozygosity. Loss of homozygosity, LOH, is that when there is an imbalance. This is a little bit less than having a big deletion mutation, which is what we usually test for, and where most of the cases are. Now, that doesn't mean that if you have that, that is necessarily causing the disease. It's going to take a long time for us to test that. This is very similar to what some of you have had on your regular mutation tests that are called mutations of unknown pathologic significance. So there is a mutation in CCM gene, but we don't know if it causes the disease. And you happen to have CCM, so you don't tell me it's not causing my disease, you know. So the way we prove that is we have to take the affected and unaffected member of your family and see if they have all of them that mutation, then it's not causing the disease in you. But if only the affected siblings have it and the unaffected do not, then we can use that mutation in your family. It's going to be something similar to that with understanding this loss of homozygosity.
Okay. And now, plasma biomarkers. We can go over if you have an extra few minutes. If you don't,
Sure, no problem.
in 5 minutes.
Yeah, no problem. So there are three types of plasma biomarkers that we've been developing, and it's extremely important to understand what we call contexts of use, because people say, well, you know, this biomarker was reported in familial cases in Italy. What does that really mean? Or this one was reported from the San Francisco group in CCM 2 and 3, and what does that mean? So that those are biomarkers of the genotype. So basically a blood test other than the mutation testing that tells you you're more likely or less likely to have CCM 1, 2, or 3. And we published a bunch of those biomarkers about 2 years ago, and including some microRNAs that talk to the genes, but yet they can be assayed in a simple blood test. That's not what we're talking about. What we're talking about with the biomarkers that I'm working on, they are diagnostic biomarkers of bleeding in CCM. So we're trying to answer the question, can you tell me if my lesion has been bleeding or not? Because a lot of you come with a headache. You didn't have an MRI when you had the headache, or maybe you had an MRI and it was negative. And we're not sure, you know, you had the whopping headache, worse than anything you've had before. You haven't felt the same way since then. And we have to talk you out of it by telling you your lesion didn't bleed. But really, we don't have all the information to tell you that. So we're trying to look at blood tests that differentiate patients who had a bleed in the prior year from those who didn't, to complement the MRI, because you're not having an MRI every 2 weeks, you know, and you come after a year and did my lesion bleed during this year, even if the MRI doesn't show it. So this is where the diagnostic blood biomarkers are. The second context of use that's very exciting, and that's the one everybody wants, is called prognostic, meaning I take your blood today, and I can tell you what's the likelihood that your lesion will bleed next year. Now, it's not going to be very high because, as I told you, even in prior bleeds, only 12% per year will bleed. So for a lesion that never bled, the likelihood that it will bleed is very, very low. But we have enough cases, thank God we have tons of patients that we study, and we've been doing it for a number of years, that we have now enough patients that bled in the next year where we had their blood before, and ones that didn't bleed in the next year, tons of those that we have their blood. And sure enough, there are blood molecules that are very different in the group that will bleed next year. And we reported this first time already about 6 years ago, and it included 2 independent samples, so meaning we did it one time and we rechecked it in another group. Now we have the ability to test it in 800 patients collected not just in Chicago, but collected at Mayo Clinic, UCSF, and Chicago. And many of you have given blood samples for that. And we're working on developing this prognostic biomarker. And the last one I want to tell you about is called the monitoring biomarker. You already heard about QSM being a good monitoring biomarker, even though it's not a good diagnostic or prognostic. If you have QSM today, I can't predict if your lesion is going to bleed. But if the QSM increased at 2 points in time, that's more likely that it had bled. So is there a change in blood tests that parallel this, so that maybe if we're doing a trial, we don't only do QSM changes, but we also look at effect on the blood biomarkers. So it's very similar to doing a diabetes study where you look at hemoglobin A1C as well as daily blood sugars, as well as heart attack and kidney damage. So of course, heart attack and kidney damage, you don't want to have too many of those. But if you affect the hemoglobin A1C and the blood sugars, then you're doing the right thing for your diabetes. So this monitoring biomarker, we're able to compare it to patients who had QSM in the TR, in the trial readiness cohort, as well as in the patients who are in our Atorvastatin trial. And we're deriving unbelievable ability to have the blood test actually parallel exactly the cases that have an increase or decrease in their QSM. So that's as a monitoring biomarker. What type of molecules are we looking at? So we're looking at some proteins in the blood. We're looking at some metabolites in the blood. And there is a unique type of molecule called microRNA that is also beneficial in those tests. However, it looks like the microRNAs are duplicated by the metabolites so that we don't probably have to use the microRNA. So you only have to assay a metabolite and 2 proteins, or 1 protein and 3 metabolites, and you'll have a 80–90% accuracy in predicting and explaining bleed. So I did it in the time we had. I'm happy to take a few questions if you want.
Oh, goodness. Okay. I wasn't screening anymore. Can these biomarkers be used to inform decisions of patients on having surgery versus monitoring the CCM?
Ultimately, yes, for sure. So, every week I have a patient that if their lesion was going to stay okay, they will do one thing. If their lesion was not going to stay okay, they'll do something else. The problem is that they cannot today because we are still validating them, and we have to test them, and we have to make sure they apply equally in men and women. They apply equally for brainstem lesions and non-brainstem lesions. So very rigorous. That's why it takes 800 cases to do the tests. But we're making very, very good progress. And I want to, give big credit to the teams at UCSF and at Mayo that helped collect patients for us and blood, as well as the many patients in Chicago. And so this will result in a letter of intent to the FDA for each context of use, just like we did with QSM. And then it's a process where the FDA will approve it or not. And then a manufacturer, a blood test manufacturing company will be the one producing the test, just like a pharma is going to be doing the trials.
I'm going to ask you one. I know there's a lot of questions in the Q&A, and I'm sorry we can't get to all of them this evening. I tried to pick them.
I may want to suggest to you, like we've always done before, if you summarize them at some point and send me an email without the pressure of answering it today, I'm very happy to share my thoughts.
Okay.
Because
But I do
definitely this is good.
I
Okay,
have
go
one
ahead.
that somebody dropped, and which I think is actually kind of cool. Do you think there will ever be a time where you can get continuous monitoring of the blood like you would with glucose monitoring? So your biomarker goes up, where you're headed.
Yeah, could that ever happen in the future? Probably, but that's not what we're after now, because it's a little bit complicated to get these metabolites assayed and having the same result every time with no error. It's hard to do. I mean, the only molecules you could do that at all times is blood sugar. That's because it's been done for years and people developed that. But to do it for tryptophan or to do it for L-carnitine or some other metabolite, that's going to be very hard to do on the fly.
Okay. All right. We'll wait till the science catches up, the technology catches up.
Exactly. That's what it is.
Okay.
Thank you so much for hosting me again. It's always a pleasure, and happy to do this as often as you think it's useful.
Okay. Thank you. And thanks everyone for sharing your time with us. The link is just in the chat for just a moment for the support group if you want to go join them. I think they're already there. And this is recorded and will be up on our YouTube channel within the next few weeks. We do closed captioning, and so it takes a little bit of time. Thanks, everybody. Good night.